normal EEV slot, but it's a bit of a special seminar in that it's given by one of the fellows of the Indiana University Institute for Advanced Study. And normally Mary Ellen Brown, who directs that institute, would make this introduction, but she can't be here. And she wants me to say a few things on behalf of the institute. And I think it all boils down for us in biology that we should maybe take advantage of this group of people a lot more. They've been here for 20 years. I've been here for 10 years, and I don't know of anyone in biology besides Joanne that's been sponsored by them. And here's the deal. They invite a fellow. They stay 3 weeks. They have a very nice office over at Poplar's. They give them some money to come and travel here and to stay here. And all they have to do is give one public lecture and interact with whoever they've come to visit. And this is Joanne's public lecture. It's been absolutely fantastic to have her here. And I would like to thank the Institute for bringing her. You want to add anything to that? So before I introduce Joanne, there's a reception tonight. Everyone's welcome. Starts at 8. Linda and I are in the corner of 2nd and Valentine. Joanne received her PhD in 1993. So 3-year PhD, is a very productive PhD. Then 1994 through 1995, she's a clinical scientist at the Communicable Disease Surveillance Center. So I'm not quite sure what she was doing there, but she was there for 2 years. And then in 95 through 97, she was a postdoctoral fellow at Oxford University, where I think she began the work that she's going to talk about today on schistosoma. Then she became a lecturer in infectious diseases at Oxford University. And then from there, she received a 10-year fellowship called the Royal Society University Research Fellowship. And she has basically what I think of as about a 10-year sabbatical grant, and where she can do research. And part of that allows her to travel. And like I said, it's great to have her here. Her title is Coevolution and Compatibility in the Snail-Schistosome System. Joanne.
Thank you. Right, as Kurt just introduced, I'm just going to talk about some of the relatively recent studies I've been doing on coevolution and compatibility in the Snail-Schistosome system. How I'm going to start is I'm going to start with just a quick general background. Is that too loud? Oh. Quick general background on the system I'm working on before going on about covering in very briefly some of the recent studies performed. Okay, schistosomiasis, or bilharzia as it's sometimes called after the person who discovered it, is a very important disease, medical and veterinary importance, second only to malaria in terms of human morbidity and mortality. There's some 600 million people exposed throughout Africa, the New World, and Asia, with some 200 million people infected at any one time with any of the 5 species of schistosomes that cause disease. Mansoni, Hematobium, Japonicum are the major ones, all of which I work on, though I'm going to talk about just Mansoni today, and Intercalatum and Mecongii, which are lesser causes of intestinal schistosomiasis across Africa and Asia. What it causes is classically shown by here. This is acute hepatosplenic disease. The problems arise not by these are worms, not by the worms themselves, but the fact that these worms live quite happily away in your body for anything from 7 to 30 years, hiding away from the immune system, pumping out hundreds of thousands of eggs every day. These eggs have little spines on them, and though they're supposed to be being passed out with feces in the urine, the vast majority actually get trapped on the way out. We have immune reaction to the eggs, causing sort of granulomas and calcification. Most commonly, they're in the liver, and it causes a severe sort of portal stem fibrosis. The whole liver is sort of calcified with this pipe stem fibrosis. By this time it gets to this stage, there's not really very much you can do apart from sort of portal shunts to release the pressure. But if you get them early enough, chemotherapy, praziquantel, is a very good drug treatment. Usually takes quite a few years to get to this sort of state in the African and the New World schistosomes because it's a very chronic disease. But the Asian schistosomes are that much more virulent because they pump out some 50 times more eggs every day. And so you get quite a few children at this sort of severe stage in quite sort of young age. Whereas most of the damage is in the liver and the spleen, you tend to get eggs trapped all over the body, and that causes a number of other acute symptoms. Particularly if you've got lots of female worms in you, you tend to get a lot of eggs trapped within the lungs, and you get pulmonary hypertension. Also, particularly the Asian schistosomes seem to have a predilection of getting in across the blood-brain barrier into the brain and cause epileptic-like seizures if it's in the brain, or lower limb paralysis if you get into the spinal cord. And also, if you live long enough, there's quite a good chance you'll get cancer in the end because of schistosomiasis. Particular Schistosoma hematobium, the urinary schistosomiasis. For example, bladder cancer in Africa is about 32 times more prevalent than it is here in the US, and that's because of the association with schistosomiasis. Yet, while these are all sort of acute symptoms, you can see people very ill, you can see people dying, the real problem of schistosomiasis is much more of a chronic thing because it is long-term. It goes on, more and more build-up of eggs in the body, and has a broader impact upon the economy and the health. For example, there's clear evidence of mental and physical retardation in children with schistosomiasis, much of which can be completely reversed on chemotherapy, on giving effective antihelminthics. And likewise, lethargy and weakness. While we may think that's not too big deal, feeling a bit tired here in the developed world, if you're a sustenance farmer or working in a paddy field, severe lethargy and weakness can have a huge impact upon their health and the economy. Seeing that its impact, even though it's sort of 600 people exposed, it's disease entirely restricted to contact with fresh water. We can see that all the schistosomes have this two-stage life cycle. We have a mammalian definitive host, where I said these worms are happily living away, pumping out lots and lots of eggs. If this person's working away, washing their clothes, or in this case, doing the paddy fields, and urinate or defecate into the water, which is actually quite common, then eggs are passed out and they hatch out in the first larval stage, which is called the miracidium. This miracidium lives about 3 or 4 days, seeks out and finds an appropriate freshwater snail. If it's lucky, successful, it penetrates the freshwater snail, undergoes asexual multiplication. From about 4 to 7 weeks later, these snails become almost like parasite production factories, and they start pumping out thousands to millions of cercaria each day into the water. This is the next larval stage, the next free-swimming larval stage. And these swim around and find another mammalian definitive host, lose a tail, penetrate through the skin, migrate through the body, and the life cycle is completed. Now, an interesting aspect of the life cycle, both in terms of the study and control implications, is that schistosomes really aren't that bothered about which mammalian definitive host they take. For example, Schistosoma mansoni really isn't that bothered. Equally successful if it infects a human, a non-human primate, or a mouse. Other ones like Schistosoma japonicum, there's over 40 species of mammal known to act as effective mammalian reservoirs of infection. This also makes control very difficult. Even if you treat all the humans and knock out the parasite for a while, it simply switches host to another mammalian host and maintains a life cycle. So it's very difficult to control. In stark contrast, schistosomes are highly species-specific at the level of the molluscan intermediate host. And a single species of schistosome can only infect a limited few or a single limited numbers of freshwater snails. And thus this is an ideal place both to study it and also in terms of hopefully developing control strategies aimed at the intermediate host stage. Yet even within these species, while they can become infected, you never can find a species of snail that can become totally infected. There's a lot of intraspecific variability too. And that's what's called compatibility. So I'll just give you the first of two definitions here, because this is what I'm going to refer to throughout the rest of the talk. We call it compatibility because we don't really know yet what's going on. It's obviously whether a snail becomes infected is the net effect of the host and the parasite interactions, and how we define it is this. In compatible interactions, the parasite recognizes, penetrates, develops within the snail, giving rise to the next infective stage, the cercaria. We can simply call the snail susceptible there. Can be infected, pass on the next stage. In incompatible interactions, the opposite. We don't know for sure whether the parasite in all cases either enters and then is destroyed, or it can't enter in the first place. But in this case, we'll call the snail resistant. And I said there's much. You go to any population of freshwater snails in the wild, and you'll always get a mixture of susceptible and resistant snails within that population. You'll never get a fixation of one to the other. And one possibility for this could be co-adaptation or co-evolution between the host and the parasite. Co-evolution, the final definition, is that which is driven by the reciprocal evolution of host resistance and parasite infectivity and or virulence. And to study co-evolution using the potential co-evolution between a host and a pathogen would have a number of implications, both from a theoretical perspective, because there seems to be a proliferation of purely theoretical co-evolutionary models arising, things like gene for gene, matching allele at multiple loci, red queen, and by actually studying what happens in the real system, empirical data will help us to evaluate these. But also in cases such as the schistosomes, we'll have an applied perspective in terms of the biomedical, understanding differences in where to predict and how it develops Snail-Schistosome compatibility may have implications such as for the evolution of host range, or even modeling how hosts and parasites may adapt to other changes in their environment, such as through vaccination and drugs. Yet, with the notable exception of perhaps some of the work here in Bloomington, in general, there's rather limited empirical research on coevolution between hosts and pathogens, particularly in animal pathogen systems. So this is what I set out to do. Snail-Schistosomes are obviously a useful and important model system in which to study coevolution. And to do it, you'd obviously have to study both the host, in this case resistance and susceptibility, and also the reciprocal effect with the parasite, virulence and infectivity, and do a combination of field and laboratory research. And the work I'm doing together with my group now is obviously covering each of those aspects, but for an hour's talk, I'm just going to summarize a few of the studies just on the side of the host, resistance and susceptibility, performed in the laboratory. In particular, I'm just going to summarize 4 studies. First 2 aiming to understand a little bit of the genetic basis of Snail-Schistosome compatibility, and then going the final 2 on how the variability and compatibility may be maintained through different trade-offs being involved. So the very first study which started it off, study to look whether compatibility is a heritable and strain-specific trait. Look at heritability because obviously a genetic basis to resistance and susceptibility is essential for coevolution to occur and for natural selection to work. And strain specificity itself is a fundamental assumption of many coevolutionary models, such as that of the red queen. So here, one very useful way in which to identify a heritable trait will be to perform artificial selection. So here, this is what I performed, using here 2 snail strains of Biflaria glabrata, one Vespiano, originally from Brazil, the second from Puerto Rico, and 2 geographic strains of Schistosoma mansoni, one again from Puerto Rico, but from a different area than the host, and the other one from Kenya. What I did was take replicate tanks of 60 snails, matched for age and size, took here all the Vespiano, exposed each snail individually to 5 miracidia here of the Puerto Rico strain. Took 5 miracidia, as that's a dose known to result in about 50% infection prevalence in these snail lines. Here in the next tanks, 60 snails individually exposed to 5 miracidia of the Kenyan strain, and the unselected controls treated identically but not exposed. And the same in the Puerto Rican ones. Then left them, started shedding them to see whether they were shedding cercaria from 4 to 7 weeks later, identifying, marking those ones whichever did shed, and then at 7 weeks splitting them into the approximately 50 within each tank that never shed within the time, and between the susceptible ones, the ones that did shed, that were infected, those to be resistant selected and those to be susceptible selected. The same for each of the replicates. Controls obviously match, reducing the number of snails within each tank to control for. I'll try down here. Could be interesting. It's where men's jackets are very good and fake pockets aren't. Okay, so to continue. So what simply did, left the parental lines to breed up until there was about 60 matched age size offspring within each tank, then exposed them to the same parasite strain to which their parents had been exposed, shed them 4 to 7 weeks later, and then checked they were of the correct compatibility status. So anybody who was selected for resistant, anybody who was found to be shedding within the resistant selected tanks were removed, and vice versa. And this was continued until the F5 generation. Well, this would hopefully indicate whether the genetic heritable trait of resistance and susceptibility. In order to test for strain specificity, the protocol had to change slightly. Here in the F3 generation, doubled the number of tanks yet again and took half the progeny from each of the F3s, exposed half to the same parasite strain to which the parents were being selected and half to a novel parasite strain to which the parents weren't being selected. And again, shed them from 4 to 7 weeks later to identify the compatibility phenotype. What was found is compatibility heritable. Well, here we can see it clearly is. This is the percentage infected. This is simply pulled the results from each of the replicates together for simplicity in this illustration, and show about 50% infection prevalence at the parental generation in unselected controls, maintained about 50% right through to the F5s. Resistance selected, on the other hand, had a clear effect, were significantly different by the F1 generation, going down to about a 25% infection prevalence to 5 miracidia at the F3 generation. Susceptible lines took a little bit longer, but significantly different by the F2, resulting in about a 75% infection prevalence with 5 miracidia at the F3 generation. Then is compatibility strain specific. Here we can see this is just the same results from the last graph presented in a different way. It's just the F3 line shown as a block. So resistance selected here, about 25% infection prevalence, 75% unsusceptible selected when exposed to the same parasite strain to which their parents had been selected, and the 50% in the unselected controls. What happens when you expose the offspring to a novel parasite strain, and you can hear a significant difference. What happens is that the infection prevalence returns to that akin to the unselected controls at about 50% infection prevalence. So here we can see heritability is also strain-specific. So the number of theoretical and applied implications of this. First of all, for the field, here we can see in the system there's potential for very dynamic situation, very fast change, evolution and coevolution. We can see the effects of resistance, selection for resistance within a single generation and within 2 generations for susceptibility. There's also implications for site-specific compatibility. For example, a snail resistant to the current sympatric parasite population, the current parasite which shares its environment, if that same snail was washed downstream during flooding or high river flows, as often occurs, they no longer will be resistant to the new parasite strain to which they're interacting with, and vice versa. And this is indeed what we see within the field. There's also a number of genetic implications. Previous work stated that a Biflaria glabrata maturity, a single gene following simple Mendelian inheritance determines whether the snail becomes susceptible or becomes resistant. Now, whilst such may well still hold true when dealing with a single host, single parasite combination, the results here suggest that we may not be dealing with this where we have more than one strain of parasite, as indeed is more akin to the field situation. Indeed, the strain specificity, where we saw selection for resistance to get to one strain of parasite, had no effect upon the resistance or susceptibility to a novel strain, hence allele frequencies, suggests that we may be dealing with a multi-locus trait here, at least a 2-loci, 2-allele model, which would give support for the matching alleles at multiple loci genetic model for coevolution. So, on that thought, leads on to the second study where I wanted to take this a little bit further in terms of the genetics involved and to see whether then resistance is a dominant trait in the system as is common within plant pathogen interactions and also other animal helminth interactions. May also explain the faster response to artificial selection and the resistance versus susceptibility. I also wanted to test whether this could be a multi-loci trait, or in particular, whereas you could artificially cross snails to be actively resistant to more than one parasite strain at the same time. So here, how it's performed looks a bit complicated, but in fact, it's quite simple, really. It's actually here took replicate tanks of about 50 or 60 individual pairs of snails. Here, pretty much continuation of the artificial selection, but using individual pairs. Here's Puerto Rican snail resistant to the Puerto Rican parasite, matched with an identical partner. Likewise here, susceptible to the Puerto Rican parasite, matched with an identical partner. And the experimental lines here are here, this group here, where you cross a resistant with a susceptible, a non-matched partner. Unselected controls, and the same going on, but within the Vespiano, the different host snail strain. What you did, removed any initial egg masses within all these lines to control for any bias in allo or autosperm storage. Bred up the F1s, I'm calling them F1s and P1s here for simplicity. They're actually the F4s and F5s from the artificial selection. Confirmed the parental compatibility phenotype, so what I'm calling resistance, checked it really couldn't be infected. And then confirmed crossbreeding occurred because these are simultaneous hermaphrodites that can choose to either crossbreed or to self. So obviously selfing would be irrelevant from such crossbreeding studies, and they were excluded from the analysis. And then took all the F1s that were left after this, which was only 14 per group, and then exposed them all to the Puerto Rican parasite, determine the compatibility phenotype. What we would predict if resistance was a dominant trait within the matched pair crosses, resistant versus resistant, we'd expect again the 25% infection phenotype, just the same as the artificial selection. Likewise, matched pairs of susceptible versus susceptible would expect the 75% infection prevalence with 5 miracidia, whereas the controls are 50. If resistance is a dominant trait, however, we'd expect in the unmatched pairs, the resistant versus susceptible crosses, in the F1s only the resistance phenotype to be shown, only the 25% infection prevalence. So that is exactly what we found. There's no difference between expected and observed, and only the resistance phenotype was shown within the F1s here between the resistant and susceptible crosses. In the second part of the study, looking to test whether we could cross snails to be resistant or susceptible to both. Here again, similar to the last one, matched pairs. Here Vespa, resistant to the Kenyan parasite, matched with another identical partner. Here resistant to the Puerto Rico with a matched partner. And here was the experimental group where one in each snail of the partner was resistant to the Kenyan strain and the other had been selected to the Puerto Rican strain. Again, unselected controls, and here the next group was the same with susceptibles, either matched susceptibles to Kenya, matched susceptibles to Puerto Rico, or unmatched pairs where one was susceptible to Kenya and one was susceptible to Puerto Rico. And the same procedure was followed. Apart from in the final stage, much like the strain specificity study, half the F1 offspring were exposed to the Kenya parasite and the other half were exposed to the Puerto Rican parasite. Again, what we would predict is, again, continuation of the artificial selection. If you're resistant and exposed to your own parasite, about a 25% infection prevalence. Whereas if you're exposed to the novel parasite due to the strain specificity, the 50% like controls, whereas in the susceptible matched pairs, 75%, or return to the 50% if it's a novel host it's exposed to. In the experimental groups, though, if we could crossbreed snails to be resistant to both parasites, you'd expect here the resistance phenotype to be shown against both parasite strains, and likewise with the susceptibility phenotype shown to both parasite strains equally. And again, it worked very well within the resistant selected crosses. The resistant phenotype was shown very clearly against both parasite strains. Slightly less strongly for the susceptible selected crosses, though still a significant effect. Slightly biased because of very high mortality in some of the groups, but it still happened. You could still crossbreed snails to be both resistant to two strains or susceptible to two strains at the same time. So, theoretical and applied implications of this. Again, we've got support. This may be a multi-loci system with the resistance as the dominant trait. And again, evidence of a very dynamic situation with very quick changes. Here we could see through sexual reproduction within a single generation, you could breed snails that were resistant, actively resistant rather than just rare, to more than one strain of parasite. But also, the converse appeared to be true. You could think it would be great for here to have a snail to breed an offspring resistant to more than one strain of parasite, but also the susceptible ones were crossing, and you'd have a snail susceptible to more than one parasite. And as this is a highly virulent parasite, you wonder why on earth they were doing this, because they seem to be doubling the chances of the offspring becoming infected and therefore dying of this parasite. So this leads us on to the third study, and another fundamental assumption of coevolutionary theory is that maybe there's some sort of cost of resistance going on. Maybe in some circumstances it's actually better to be susceptible to infection than to be resistant. Indeed, the theory predicts that for there to be a stable genetic polymorphism and disease resistance, as indeed we see in wild populations. As I said, we never get a fixation of resistance in wild populations. We always get a mixture of susceptible and resistant snails within any wild population. The fitness of the resistant genotype should be less than that of the susceptible genotype in the absence of disease. While this is brought into much theory, there's very few solid, convincing empirical evidence of demonstrating what the cost of resistance may be in an animal pathogen system. Nevertheless, saying that and seeing this strange effect, I thought it was worth looking for. So again, using the same artificially selected line, started at the beginning for resistance and susceptibility, I decided to look for potential costs here in terms of snail fertility, defined here as the actual reproductive success, measured as the number of viable offspring produced, and also in terms of snail fecundity, as can be defined as a potential reproductive success, the number of eggs or embryos produced by each of the snails. So again, the experiment performed in two parts. And the first experiment recorded at weekly intervals in each of the tanks, each of the replicate tanks of the artificial selection, in each of the generations, 5 parameters. The number of adult mortality, the number of egg masses produced, the mean number of eggs per egg mass, any malformed or mutated egg masses produced, and the number of viable offspring produced in each tank. As obviously as such a correlational study with artificial selection will have a number of confounding variables, because inherent within the design, everybody within, all the snails within the resistant selected tanks were uninfected but exposed, controls were uninfected and unexposed, and susceptibles were all infected. They performed a second part of the study using individual F5 snails from in individual isolation pots from the artificial selection lines. Here in the first group had susceptible selected infected, in the second susceptible selected uninfected and unexposed. Third group had the unselected controls infected, fourth group unselected controls uninfected and unexposed, and the last group had the resistant selected snails uninfected. Ideally, I would have also had a resistant selected infected, but resistance was getting pretty good by then, and I only had a sample size of 2, so removed it from analysis. What was found in the first part of the study, here we can see adult survival, clear across generation, mean percentage of snails surviving, a clear significant difference by the F2 generation with the lowest mortality, the lowest survival within the susceptible selected lines. Here, because most of them were getting infected and they were dying of infection, was no difference between the controls and the resistance across time and generation. When you look at fecundity, the egg production, there was much scatter. There was no difference at all at any generation or between any of the lines in the number of egg masses produced, nor the mean number of eggs per egg mass, nor the number of malformed egg masses between the susceptible control and the resistant lines. Nothing at all happening. However, when you look at fertility, the actual number of viable offspring produced, there was a clear effect, where here, by the F2 generation, was significant difference with the highest number of offspring produced within the susceptible selected lines, followed by the unselected controls, and significantly lower offspring produced within the resistant selected lines. These results were very much complemented by the second part, the experiment 2. Here we can see over the weeks, the mean percentage of snails surviving, there was a clear effect of infection, where much higher survival within the uninfected groups versus the infected groups, but no difference at all between the snail lines. Basically, the parasite alone was killing them rather than difference between the lines. Again, like in the first study, there was no difference at all in any fecundity measure. They were still pumping out the same number of egg masses, mean embryos per egg mass, but a clear difference in the viable offspring being produced. Here, the highest in the susceptible, followed by the controls, and the lowest in the resistance, with absolutely no effect of infection status. This was effect of artificially selected line, but not infection status. And so, theoretical and applied implications here. Again, what could be happening, what sort of the general assumption could be that for such a cost, a clear cost here, one of the first demonstrations, there could be an energetic trade-off. You may automatically think that snails are pumping all their energy expenditure into a defense rather than reproduction. But that's obviously not going to be a sufficient explanation here because the fecundity was matched between lines, so the energy expenditure was the same. Instead, there seemed to be something that stopped the eggs being laid actually hatching into viable offspring. Perhaps resistant snails are deficient in some internal factor such as albumin gland functioning or essential nutrients that's used both for fertilization of eggs but also essential for normal parasite development. What may also be involved is some sort of form of genetic trade-off where the genes for both fertility and for resistance are linked by simple virtue of where they're being placed on the genome, where selection for one will have result in indirect negative selection for the other and vice versa. In terms of functionality, again, such a cost will help explain why we get such polymorphism, such variability maintained within natural populations. There's always going to be a trade-off in these snails between the cost of susceptibility in terms of this increased mortality if they're infected and cost of resistance through this decreased egg fertility. And this will give a clear advantage to susceptible snails when there's no parasites around, which may account for some of the infection epidemics. For example, in places like Senegal, where through industrialization changes, vast expanses have turned from salt water to fresh water, snails colonize, and these snails that colonize appear to be highly, highly susceptible. You can take them to a laboratory and you can get sort of 80–90% infection prevalences. What happened in the 1980s, a very few cases of parasites got introduced to this area into a hugely susceptible snail population, and the resulting huge disease epidemic in the human population of Senegal, enhanced by the lowered acquired immunity exposure within the humans. Likewise, all over places with schistosome infection, such a susceptible advantage can also have implications, because wherever, for example, humans are treated with chemotherapy with antihelminthics, the parasite population going into the environment will temporarily go down. Therefore, we may get a boom back, an advantage to susceptible snails. So if there's any sort of lax in keeping of the chemotherapy going in the humans, and people then reintroduce parasites to the population, again, we'll have a highly susceptible snail population and concurrent risk for high human levels of epidemics. But we can also, in some ways taking it back to the theoretical side, then seeing if we have such a profound cost of resistance in terms of reproductive success of this snail, why on earth are we maintaining resistance genes in the laboratory at all? Some of these snails will be kept in laboratory 25, 50 generations without any exposure to parasites, but it's clearly they're maintaining the resistance genes to allow the artificial, as shown by the rapidity of artificial selection for resistance to occur. So it could be there may be some other benefits of a snail being resistant going on, quite independent of infection status, something such as potentially differential mating success or strategies. So this leads us on to the final part of the talk, looking at if there is such a trade-off, and here decided to look for, in particular, if there was differences in the sexual strategies between snails associated with a compatibility status. This is because the role of parasites on host reproductive strategies has gained a lot of interest, and there's a lot of studies trying to link current infection status on aspects such as mate choice, but there's very limited studies looking at potential infection status on mate choice in terms of a host's genetic predisposition to parasite infection at some point in the future. It may well predict that this would be a good thing, quite likely to happen within the Snail-Schistosome system, though they can't get resistance genes for themselves through sex like bacteria can. Discrimination would produce a better genome-environment match for the offspring than would random mating. So here, again, perform the study in two parts. First, a molecular analysis of reproductive strategy, simply looking to see if there was any difference in the propensity of this simultaneous hermaphrodite to outcross or to self, depending upon its genotype. Simply took F5 and F6 snails from the artificial selected lines, 60 matched pairs of susceptible selected, 60 matched pairs of resistant selected, and 20 matched pairs of unselected controls, and left them to breed. Simple as that. Took the offspring and the parents and identified through molecular analysis who crossed or who selfed. What was found within the unselected controls, 100% of the progeny were the result of cross-fertilization. And this is consistent with previous work on this snail, or unselected, uninfected lines of these snails. They like to cross. When looking at the selected line, the susceptible selected, also 100% of the progeny were the result of cross-fertilization. This just shows your rapid gel crossbreeding families. Here you can see one parent has this banding, other parent has this banding, and the offspring here have a combination of the banding patterns of both parents, indicative of crossbreeding. When you looked at the resistant selected, on the other hand, whereas 74% of the progeny were the result of cross-fertilization, 26% of the progeny were found to be the result of selfing. And this is just another rapid profile here. We can see the offspring have the banding pattern of one parent only, indicative of selfing. So see that resistance genotype does affect reproductive strategy, resulting in a mixed mating system where we see both a combination of outcrossing and selfing going on. Wanted to look at this in a little bit more detail to see what's exactly going on, and then complemented this with a behavioral analysis of the reproductive strategies. And here, took my snails again from the artificial selected lines, replicate tanks, took a susceptible selected, stuck on some super glue and a long piece of string to one side of the tank, resistant selected stuck to the other side, so they freely can move about on each side of the tank but not come together to mate, and had a free unselected control who could go around and mate with one or both, or not simultaneously. 20 trials where the control was uninfected and 10 infected with the same parasite to which these had been selected to. And the entire behavioral repertoire of these snails were observed continuously over 8-hour nights by me and one of my master's students, which was lovely. And what was found, well, to begin with, looked like nothing at all. Snails, whether they were infected or uninfected, mated just as much as each other. There was no difference in the duration or the incidence of mating, whether you were infected or not, nor whether they mated with a resistant or a susceptible. Didn't seem to be bothered at all who they mated with. When you're looking, when the free-moving snail was uninfected, and look at gender, here being a simultaneous hermaphrodite, again, absolutely nothing was happening. This is where the pink refers to the free-moving snails mating as female or as male with a resistant snail or with a susceptible, and absolutely nothing going on. However, when the free-moving snail was infected, clear mate choice went on, where the resistant snail actively refused to be mated in the female gender with an infected partner. And no matter how often the infected tried to insert its penis, it just got its penis bitten and termination of sex. So it accepted to mate twice as long in the less preferable female gender. So here we can see again resistant genotype does affect reproductive strategy, resulting in the mixed mating system, but that it's displayed through a combination of both resistance genotype and current parasite pressure, and it's displayed through the more subtle effects of gender biases rather than total mating activity. So this could raise a number of implications, such as for the evolution of sex. For example, it may reflect the cost-benefit ratios of outcrossing to selfing. Particularly in the molecular study, you saw the propensity to outcross depended on the propensity that you'd be infected by a parasite at some time in the future. And thus, all the susceptible selected and all the controls outcrossed, whereas the resistant selected and the only ones less likely to become infected if exposed in the future also performed partial selfing. They thus get both the advantages of sufficient outcrossing for recombination, for sufficient variability to any co-evolving parasite in the future, but also through selfing may avoid outbreeding depression through the breakup, through recombination of successful co-adapted gene complexes, in this case for resistance. And how they do this may reflect the differential benefits or costs of the genders. Male genders in most species, and also snails, is less costly than mating in the female gender, so resistant snails refused to outcross in the female gender, thereby avoiding contaminating their particularly costly female egg production, particularly due to the high costs of resistance in terms of egg fertility shown by the previous study. So the implications may also have some of this for recognition systems, because here we saw in these snails clear evidence of recognition and discrimination, both on the partner's infection status and potentially upon the host's own compatibility genotype. And how they do this may have been occurring during the extensive grazing periods of the slime on each other's shells that preceded every sexual act. And if so, the similarity to mate choice through odor cues in humans and mice on the basis of AMHC through odor may be apparent, and may favor the idea that complex recognition systems such as the human MHC may have evolved from these conceptually simple but obviously highly efficient invertebrate immune defense systems. So to sum up, hopefully just have a taste of these 4 studies should highlight the applicability of snail-schistosome interactions for studying of coevolutionary theory, and also their importance both for sort of evaluating control strategies and predicting disease dynamics, most clearly seen with things like the susceptible advantage, and obviously in association with the ongoing work with obviously the reciprocal work upon the schistosome variance and infectivity, and both comparing definitive hosts with intermediate host dynamics, and of course bringing this out into the real world where parasites and hosts rarely, if ever, live in isolation. So a number of studies also involved at the moment looking at multiple host, multiple pathogen genus, species and strain interactions. So finally, just like to acknowledge Mark Woolhouse, who I started the Snail-Schistosome work, Joe Hoffman, who was a master's student that helped me watch snails all night, and Manuel Berda, who also helped with the analysis of the snail videos, Kurt Lively for allowing me to come here and joining his group, the Institute of Advanced Study and the Women in Science program for sponsoring me, my funding bodies, and the rest of my group, though not involved with the studies presented here today. I just provide a great environment. Thank you.